In this lecture, we're going to go through the basal ganglia. I'm going to tell you some lies, and we're going to pretend like there's a fairly linear pathway or pathways through the basal ganglia. But like I just told you, I'm going to lie to you. It's not really true. The basal ganglia are interconnected with one another. So even though we're going to talk about a projection from the striatum to the GPE, it goes both ways. The GPE projects back to the striatum. So it's a bit of a mess. But we'll do what we can to make sense of it. In the first part, I'll go through the nuclei of the basal ganglia. Then I'll outline the main pathways. We'll talk about the effects of dopamine on these pathways. And then we'll present one of our current models for how we think the basal ganglia allow us to select appropriate actions. Because that's their function. The basal ganglia are a collection of subcortical nuclei that allow us to execute appropriate motor programs. As such, they're going to receive input from a few different sources. How do we know what's correct? Well, of course, the cortex has to tell us what are we planning on doing. So we have to have input from motor cortices. We also need to know what are we doing? What's currently going on? Where am I? So we need input from sensory and association cortices. We probably need input from emotional circuits as well. So we're going to get diverse input to the basal ganglia so that they have a good idea of what's going on. Now, they don't get sensory input from the thalamus or anything like that. They get our perceptions from the cortex, so fairly high level information. What do we think is going on, in other words? There will be input from the thalamus, but that's going to be from the intralaminar nuclei, and that's just going to diffusely stimulate the striatum. We're not getting sensory input. We are getting our perceptions, though, and we're getting our plans. And that's what the basal ganglia needs to know in order to determine, is this the right thing to do? So we'll be going through all of these different connections in this talk. It looks like a mess, because it is a mess, but we'll try to make sense of it. We're going to start off in the striatum. So this is the major input nucleus. It's fairly massive, and it's going to be inhibitory. We can chop the striatum up into a couple different nuclei. So there's the caudate and the putamen. The caudate is going to have a head and a tail. It's going to run a little more medial to the putamen that's a little more lateral. That doesn't really matter. This is where we're hanging out in this lecture. We also have the nucleus accumbens. The nucleus accumbens is a little more ventral, sometimes called the ventral striatum. This deals more with reward and emotion. The dorsal striatum deals more with what are the motor programs that we're going to execute. So we're going to stick in the dorsal striatum. So if you look at these data here, they're showing you the various types of input that the striatum receives. So in the upper left, we have the cortex, and there's a color-coded map showing you the projections. On the top right, you can see the intralaminar nuclei. So none of the relay nuclei, just the non-specific intralaminar nuclei. And down in the middle, at the bottom, we see the striatum. There's the dorsal striatum that we'll focus on today. This is going to get input from the motor regions of the cortex. So in the dorsolateral striatum, we have a body map. Just like we have in all of our other sensory motor regions, there's a place for the head, a place for the forelimb, a place for the trunk, and the hind limbs, of course. We have a map of our body that allows us to then select certain actions related to those parts of the body. The nucleus accumbens, or the ventral medial striatum, this is going to get input more from limbic regions. So hippocampus, amygdala, for example, as well as visceral afferents that are related to the emotional state. So this will allow our emotions and our memories to affect behavior. Where we're going to hang out is the dorsolateral striatum. And there's an intermediate zone, of course, that's kind of between the two. Here it's not so much motor plans, it's more associational cortices relaying our perceptions. Now within the striatum, No matter where you are, we have about 4 cell types. If you had to know only one, I would hope it would be the spiny projection neurons. They're exactly what they sound like. They're neurons that project out of the striatum, and they have dendritic spines. You'll sometimes hear them called medium spiny neurons, and that's okay. That's an older name we're kind of getting away from. Now we call them the spiny projection neurons. These are GABAergic, just like almost everything else we're going to talk about today. We also have the large A spiny neurons and the small and medium A spiny neurons. So in the striatum, spiny projection neurons are going to account for anywhere between 90 to 95% of all the cells there. That's why if you had to know one, this would be the one. Spiny projection neurons are going to project along a couple different pathways, but these are going to be the output. It's going to be GABAergic, so we're going to be inhibiting whatever they project to. We'll draw these out in just a bit. Local interneurons are going to be the large A spiny neurons. These are special because they release acetylcholine. The small and medium A spiny neurons, these release GABA. Now, spiny projection neurons target other spiny projection neurons to provide a little bit of negative feedback. So in this case, if we have 2 SPNs, whichever one is active will inhibit its neighbor. This is just some lateral inhibition that helps focus activity. Because we're going to get kind of diffuse excitatory input from the cortex, and to help focus that, whichever neuron is active, make sure to inhibit its neighbors. So this activity stands out more so than any little noise. Spiny projection neurons are also targeted by the small and medium A spiny neurons. And they're going to be the targets of our large A spiny neurons. Now this could be excitatory or it could be inhibitory. It depends on what kind of muscarinic acetylcholine receptor they have. So what we're going to find here is that our SPNs are going to have one of 2 types. M1, which is GQ coupled. The odds are GQ, so M1, 3, and 5. Or they have M4, GI. So the evens are GI coupled. Excitatory or inhibitory. And we're gonna soon find out that we have 2 populations of spiny projection neurons that can be separated based on their dopamine receptor content and their acetylcholine receptor content. In our interneurons here, we have nicotinic acetylcholine receptors. So the large A spiny neurons are going to stimulate these small and medium A spiny neurons that then inhibit spiny projection neurons to decrease their excitability. Here we can see spontaneous, inhibitory, postsynaptic currents recorded in what they're calling medium spiny neurons, but we know to call spiny projection neurons. And what they're showing you would be the large A spiny neuron on top and the spiny projection neuron on the bottom. These iPSCs are recorded from input from the small and medium A spiny neurons. So what we're looking at is how much inhibition do the spiny projection neurons get. So on top, no activity of the cholinergic neuron. If you go to A2, you'll see that they're stimulating the cholinergic neuron there, and when they do that, there's an increase in the amount of inhibitory currents. So these cholinergic neurons are stimulating inhibition of spiny projection neurons. They are going to stimulate excitation, though, in some spiny projection neurons. So they provide some diffuse inhibition via these interneurons, and then they're going to allow for selective excitation based on the muscarinic receptor that's expressed. And we'll go through these around the end of this talk whenever we put together all of the basal ganglia. So the SPNs are going to have their activity shaped by the A-spiny neurons. It's a little complicated with the lands. The small and medium A-spiny neurons just inhibit. That's pretty straightforward. And the spiny projection neurons provide a little bit of an intrastriatal collateral here to inhibit one another as well. So there's your striatum, major input nucleus. We're going to go ahead and start to build our basal ganglia. So the cortex is going to be our input. Now this is always communicating with the thalamus, what we call the thalamocortical loop. Now we're dealing with motor function, so the ventral anterior and ventral lateral nuclei of the thalamus are going to be the principal targets here that we're concerned with. So excitatory input from the cortex, excitatory feedback from the thalamus. How much excitatory feedback? Well, that depends on the basal ganglia. So, dorsal striatum. Major input nucleus from the cortex. Another input nucleus is going to be the subthalamic nucleus, and that's below the thalamus. So we'll put it below the thalamus. The STN also gets input from the cortex. Now the STN, like everywhere else, has a body map. So it has a place for the head, the forelimb, the trunk, the hind limb. And it's going to get input from the cortex. It'll get input from eye fields to help us generate saccades. It'll get input from the primary motor cortex, the supplementary motor area, and the premotor areas. Unlike the neurons in the dorsal striatum, unlike the spiny projection neurons, that is, STN neurons pacemake. So these things are always active to some degree. In the striatum, our output, which we'll draw in just a little bit, silent. No pacemaking. The subthalamic nucleus is going to project to our output nuclei of the basal ganglia. So, we're going to have glutamatergic projections here. That's what's special about the STN. These are the only glutamatergic neurons that we're going to find in the basal ganglia. So we have our output nuclei, the internal globus pallidus and the substantia nigra, pars reticulata. These, of course, have a body map as well. The GPI is going to have our forelimbs, trunk, and hind limbs, while the SNR is going to have the face and mouth. The STN is also going to project to another nucleus here, the GPE, which will be targeted by the striatum, as will the output nuclei. So we're starting to build the mess a little bit at a time. So, we've gone from cortex to striatum, that's going down the right there. We're also going from cortex to STN, so there's our two inputs, the striatum and the subthalamic nucleus. The outputs are going to be the internal globus pallidus and the substantia nigra, pars reticulata. These inhibit the motor nuclei of the thalamus. Everything's inhibitory except for our STN. So, here in the GPI and SNR, we're going to get focal input from the striatum. This thing's massive. The STN, which is small, this is going to provide more diffuse input. And we'll circle back to this in the end, whenever we put the basal ganglia together. So, the striatum is going to selectively inhibit the output nuclei to selectively disinhibit the motor nucleus of the thalamus. That'll allow us to select specific actions, while the STN is going to grossly excite the GPI and SNR to grossly inhibit the thalamus and prevent action. Here, GPI and SNR, like everywhere else except the dorsal striatum, going to pacemake. That's what that sign tells us. So we got to consider the different polarity of our input, as well as whether it's focal or diffuse. Almost done here. The external globus pallidus is going to provide a kind of diffuse input to the STN and the output nuclei. These also pacemake. So everywhere pacemakes except the spiny projection neurons in the dorsal striatum. Everywhere is GABAergic except the STN. Now the GPE also provides some feedback to the dorsal striatum. Like I said, it's a bit of a mess, but we're gonna leave that off here, tell a little old white lie, and try to put our basal ganglia together. One last place to consider, and then we've introduced all of the nuclei, and that is the SNC, the substantia nigra pars compacta. These pacemake. What's special about them is that they release dopamine. Dopamine is going to target the rest of our basal ganglia, but the site that we care about is the dorsal striatum. These are going to target our spiny projection neurons, and they're going to do so differently, depending on whether they're along the indirect or direct pathways. And that's because they have different dopamine receptors. The direct pathway that we'll walk through in just a bit, those have D1 dopamine receptors. The indirect have D2. Now these dopamine projections, these are massive. We don't have dopamine neurons everywhere. There's only a couple of places. There's the SNC and the ventral tegmental area. Those are both in the midbrain, and they've got to supply the rest of the nervous system with all the dopamine that they need. So the axons of our dopamine neurons are going to cover fairly large terminal fields. That's what the data here show us. So there's a single neuron in the SNC that they've traced. So if you look at the red and black traces, that's just showing you an axon. It branches and hits a couple different areas. The red one and the black one are going to both innervate the dorsal striatum, that CPU, caudate, putamen, that's another thing, another way of calling it the dorsal striatum. Same thing. So these dopamine neurons cover a very large area. They got a lot of axon, that means they have a lot of ion channels. That means they're going to have a lot of metabolic stress. They're also always firing action potentials. Unfortunately, they use calcium to pacemake, not something safer like sodium. So, they're prone to die. Dopamine neurons die. They will die in all of us. Luckily, because of that large terminal field, we won't notice until we lose about half or more of our dopamine neurons. When that happens, we're going to see a hypokinetic state called Parkinson's disease. So now we've built our basal ganglia. Let's see if we can walk through the pathways and allow us to execute an action. Now, there's two main pathways that you need to know. We'll introduce a third, but you only really need to know two. Two is enough to confuse you. We have our direct pathway and we have our indirect pathway. So the direct pathway we'll put in black. For the direct pathway, our spiny projection neuron is going to become activated and inhibit the output of the GPI and SNR. That decreased inhibition from the direct pathway allows for excitatory feedback from the thalamus. Thus the cortical spinal tract is active and we do stuff. So for the direct pathway, go. The indirect pathway, a little longer, but we'll get there. Like the name suggests, it's indirect. So we're not going directly from the striatum, the input, to the output. We're going to make a pit stop. So the indirect spiny projection neurons are going to inhibit the GPE. GPE is less active, so it doesn't inhibit its targets. Thus the STN is more active. The output nuclei are less inhibited by the GPE and more excited by the STN, leading to an increase in basal ganglia output. That inhibits the thalamus, so we don't get as much excitatory feedback, and therefore the corticospinal tract is not stimulated. No go. There's your direct and indirect pathways in a nutshell. Dopamine is going to have different effects on the direct and indirect spiny projection neurons because they express different dopamine receptors. Here we're looking at a single cell PCR from direct on top and indirect on bottom spiny projection neurons. And what you'll notice on top we find substance P, fine, but it doesn't really matter here. And we also find D1 dopamine receptors. You probably recall that D1 is GS coupled, D2 GI. So the indirect has a GI coupled dopamine receptor while the direct has a GS coupled dopamine receptor. These different populations then project along different pathways that have different effects on motor function. What the direct pathway is going to do is stimulate movement. That's its job. So, the direct spiny projection neurons disinhibit the thalamus so we can do stuff. Now when there's an imbalance, where we have hyperactivity of the direct pathway compared to the indirect pathway, we see an excess of movement. And that's what we see in Huntington's chorea. So in Huntington's disease, indirect spiny projection neurons are lost. Because of that, that shifts our balance toward direct pathway. And we see involuntary movements as a result. Chorea means dance, so we see kind of dance-like movements. It could be in the limbs, head. It will eventually spread to affect the entirety. But direct pathway promotes movement. Indirect pathway prevents movement. When we lose the indirect pathway, we have involuntary movements. We're not properly filtering out the noise. What we have to keep in mind is that there's a lot of noise in our cortex. And the basal ganglia acts as the filter. We have a bias toward indirect pathway activity. We must relieve it with a little burst of dopamine in order for us to do stuff. Whenever we have the loss of indirect pathway neurons that are normally going to be a little more active, we see an excess of movement. The loss of indirect is basically the same thing as an increase in direct, so we see go. On the other hand, whenever we see a bias toward indirect pathway activity, we see a poverty of movement. And that's what we see in Parkinson's disease. So this gentleman's going to try to stand up and walk around. It'll take some time. In Parkinson's disease, loss of dopamine neurons in the SNC is going to prevent inhibition of the indirect spiny projection neurons. It's going to decrease stimulation of the direct spiny projection neurons, and thus we get a shift toward indirect pathway. No go. What we see a lot more of is inhibition of the GPE, disinhibition of the STN and the output nuclei. That leads to an increase in inhibition at the thalamus, and the initiation of movements becomes difficult. The swift execution of movements becomes difficult. So we see akinesia, hypokinesia, and bradykinesia. And these are typical traits of Parkinson's disease. So there the gentleman just had a bit of a stumble because he was unable to initiate his next step. He's biased toward no-go. So everything seems like a bad idea. And there's also a hyperdirect pathway. It's going to run from the cortex to the STN. What the hyperdirect pathway does is stimulate the STN. That, of course, then stimulates the output nuclei. Now it's hyperdirect, so I'm going to put it in front of the direct. That leads to inhibition of the thalamus. So, hyperdirect is going to come before direct. And we think of this as basically cutting down the noise that's going on in our motor nuclei. So just kind of non-specific silencing. And so when you have damage to the STN, which is going to affect both the indirect and hyperdirect pathway, you see a lot more noise. So you'll see large amplitude involuntary movements. Melismus. So if it affects one side, hemimelismus. Here we can see a gentleman with hemimelismus. So damage to the subthalamic nucleus affects the activity of the hyperdirect pathway, as well as the indirect pathway, but a little more specifically the hyperdirect. Right? We still have this inhibition, this disinhibition from the GPE for the indirect. Damage in the STN, affecting the hyperdirect pathway, leads to an excess of movement, so we're unable to filter out noise. So what is dopamine doing? Dopamine is going to act in the dorsal striatum to allow us to swiftly execute actions. So we have our D1, D2 receptors on different populations of spiny projection neurons. D1 is GS coupled. We increase cyclic AMP. Cyclic AMP stimulates cyclic nucleotide gated cation channels. So we have an increase in cation conductance and thus depolarization of DSPNs. The GI coupled D2 dopamine receptors. Those decrease cyclic AMP levels and thus decrease cyclic nucleotide gated cation channel activity, leading to hyperpolarization or the inactivation of ISPNs. So we have kind of a baseline indirect spiny projection neuron activity, low level of direct. When we get a burst of dopamine, direct pathway becomes active, we go, and then we restore our bias toward inaction. Because the general function of the basal ganglia is to allow us to execute appropriate motor programs swiftly. When we see improper activity in the basal ganglia, we see inappropriate movement. So we saw some human examples. This also works in mice as well. So when you lesion the basal ganglia, depending on where the lesion is, you'll see different types of changes. So a lesion in the SNC creates Parkinson's, a lesion in the STN creates hemimelismus, a lesion in the striatum, if it's specific to the indirect pathway, can create Huntington's disease. Whenever you change the amount of striatal output, you change the amount of movement. So in general, decreasing striatal output leads to bradykinesia. An important aspect of striatal output is to relieve that tonic inhibition of the thalamus, because remember, these pacemake. So the output nuclei are constantly dousing the thalamus in GABA. It's only through the direct pathway that we inhibit the output and disinhibit the thalamus. So if we want to go, we need striatal output. When we decrease striatal output, we see a decrease in activity. It's slower. When we see an increase in striatal output, just gross elevations in striatal output, not specific for one pathway or the other, we see involuntary movements. And that's what the data over there are showing us. So this IEM1460, this is an AMPA receptor antagonist. It's going to be specific for those small and medium A-spiny neurons. So the local inhibitory interneurons in the striatum. They're also called fast-spiking interneurons. Of course, we have to have lots of names for the same thing. But what you can see here is when they apply the drug, so if you look in the top left, the excitatory postsynaptic currents in fast spiking interneurons, shown in red, drops. So we're decreasing the activity of our fast spiking interneurons. The top right shows you the actual spiking. We see a decrease in the spiking in red there. The blue is showing us the spiny projection neurons, also called medium spiny neurons. You'll notice they increase their activity. And along with this, we see dyskinesia in the mouse. We see involuntary dystonic states. So an increase in involuntary motor output. So these mice have involuntary movements that kind of track them in different positions. In Huntington's disease, what we see is the loss of the GPE targeting enkephalin expressing indirect spiny projection neurons. So the top row is showing you control. We're looking in the GPE on the left, and we're looking in the GPI on the right. So GPE on the left, GPI on the right. Indirect on the left, direct on the right. If you compare the fibers in the middle and bottom row, these are from patients with Huntington's disease. You'll notice the left has a much greater decrease. So in Huntington's, selective loss of indirect pathway spiny projection neurons disinhibits the GPE, so it's more active. That inhibits the output nuclei a whole lot more, and thus we see an increase in motor output. So we see greater disinhibition of the thalamus, and we see involuntary movements, just like we saw a few slides ago. In Parkinson's disease, we have the loss of our dopaminergic neurons, the SNC. So here's the target for PD. Here's the target for Huntington's disease. And in both cases, we're seeing the loss of these neurons. Now, the function of dopamine is to cause a switch from indirect to direct, and then back. So we get a burst of dopamine, we get a burst of direct, and we do stuff. In PD, the loss of dopamine makes us unable to get that burst of direct pathway output, so we can't initiate movements. And that's what explains the bradykinesia and akinesia in Parkinson's disease, just like we saw a few slides ago. Now, that bias toward indirect pathway activity has to do with what those large A spiny neurons are doing. So let's get those back on the board. The large A spiny neurons are going to inhibit our direct spiny projection neurons, which express the GI coupled M4 acetylcholine receptors, while the indirect spiny projection neurons will be stimulated because they have the M1 GQ coupled muscarinic acetylcholine receptors. So the tonic output here of our large A spiny neurons, these things pacemake too, keeps the D1 direct spiny projection neurons inhibited and keeps the indirect spiny projection neurons a little stimulated. So here you can actually see the pacemaking of large A spiny neurons. So whenever we want to initiate a movement, quickly that is, we need to have inhibition of our large A spiny neurons, and that's what we see. So after monkeys are given a cue to move, that's that trigger signal at time 0. The activity of large A spiny neurons tends to decrease. So after that trigger signal, you'll notice there's a little bit of blank there. So what we're looking at is just raster plots. So multiple trials on each row, and all the little ticks are showing us activity in large A spiny neurons. Once they deliver the trigger, there's a pause period, and then the pacemaking resumes. So the trials line up just like this. So you follow your rows. If you look after the trigger, you'll see a pause in your large A spiny neurons. at least on the left. So in the left, the monkeys weren't given a heads up. They just received the trigger and they had to quickly execute the action. On the right, they gave a little bit of a heads up. So you see a little less decrease in large A spiny neuron activity. But when you want to get something done in a hurry, you need to be able to robustly stimulate your direct spiny projection neurons. Dopamine may be helping us here. Not only does it act on the spiny projection neurons, it's also going to inhibit the LANs, because the large A spiny neurons have D2 dopamine receptors. And there it is. What could be simpler? As long as you're willing to lie about it, the basal ganglia make a whole lot of sense. So let's walk through and see if we can put together our hyperdirect, direct, and indirect pathways. So, what we think is going on is that a balance of direct and indirect pathway kind of biases us toward more or less activity. In dopamine depleted states, like Parkinson's disease, where we see an increase in indirect pathway activity, we see hypokinetic symptoms. In Huntington's, where we see loss of indirect spiny projection neurons, where we have more direct than indirect, we see excess movement. You can also see this in people who use cocaine. So cocaine is going to increase dopamine levels, biases toward direct spiny projection neuron activity, and boy are they animated. They're moving all over the place, they love to talk, and that's just the basal ganglia inappropriately filtering their actions. So the balance in direct and indirect pathway can explain these differences in PD and HD. If we want to bring the hyperdirect pathway into account, then we need to think about timing. So the hyperdirect pathway is going to take 5 to 8 milliseconds or so to affect thalamocortical signaling. Pretty darn quick, in other words. That's why it's called hyperdirect. It's more direct than direct. And we see general inhibition in the thalamus. So, I like to think of the hyperdirect pathway as shh. It's just going to quiet down activity. We have input from the cortex to the STN, diffuse excitation of the output, and diffuse inhibition of the thalamus. Shh. I don't care what you're talking about, just be quiet. Then the direct pathway comes in, 15 to 20 milliseconds at this point. Cortex to striatum, striatum to output, so we inhibit the output, but remember, it's focal. We're going to have selective targeting of output neurons, so we have selective disinhibition of the thalamus. So hyperdirect, shh, direct, do this thing. Then the indirect pathway comes in, about 15 milliseconds later, so we're at 30 milliseconds at this point, to turn off that motor program. So we inhibit the GPE, that disinhibits the output, and the STN, which excites the output, and we then inhibit the thalamus. So, hyperdirect, shh, direct, do this one thing, indirect, stop. So if you're kind of a fan of thinking about things in 3 dimensions, so the hyperdirect pathway is going to decrease thalamocortical signaling. That's what that pit is that it's creating there. So we're decreasing thalamocortical signaling. Grossly though, remember, these are diffuse. So gross inhibition, selective disinhibition with the direct pathway. Do this motor program. Then the indirect pathway comes in to again quiet the whole mess. And we're done. And we're also done with delivering content in this class. If anything from this lecture wasn't clear, use the questions box to let me know. I'll see you later.